hoş geldiniz. Sabah erken saatlerde başlamış oluyoruz. Ben tabii çok güzel bir başlıkla bugün güne saat 8'de başlayanlarımız var. Biz de devam ettirmiş olacağız. Size Dr. Tonya White'ı kısaca tanıtmak istiyorum. Dr. Tonya White received a bachelor's degree in electrical engineering from the University of Utah and a master's degree in electrical engineering from the University of Illinois. She obtained her medical degree from the University of Illinois. Thereafter, she completed a combined residency in pediatric psychiatry and child and adolescent psychiatry at the University of Utah and a research fellowship in neuroimaging from the University of Iowa. She was an assistant professor at the University of Minnesota for 8 years prior to joining the faculty of Erasmus University Medical Center. She started a PhD in the Department of Biomedical Engineering at the University of Minnesota and then completed her PhD at Erasmus University. And we will be listening to her with her great experience, and I want to say welcome again, please.
Thank you very much, and I'm very happy to be invited and to have the opportunity to come present some of the work that we've been doing at the Erasmus University in Rotterdam. So I'm someone from America who moved to the Netherlands, so I understand some differences in language. So if there's some things I am not saying clearly, please feel free to ask and interrupt. This is a Meet the Expert session, so it seems very formal with a long haul, but if you have a question, I don't mind being interrupted. So, first of all, I have no disclosures related to the pharmaceutical industry. I do have a number of grants, and I'm also involved in journal duties and on one scientific advisory board, but nothing that I say today should have any conflict of interest. But what I am going to talk today is about a very, very important period of our lives that all of us experienced, although we don't remember it. And this notion of what happens during the first 9 months of our lives and what an impact that can have on the rest of our lives. And I'm going to be focusing on the brain and to be giving some information from our studies on how pregnancy and that period in pregnancy can shape brain and behavior. So I'm going to start by giving 3 slides, which I'm going to point out some key features of these slides. They're busy slides. They have a lot of information, but I want to point out some important aspects of this. And I hope you all can see it, and I don't know if my mouse can work. Yeah, here. So this is borrowed from Chuck Nelson, and it's brain development over the lifespan. But if you look at this, I want you to notice on this image, this is 1 year of age. So about half the slide here is devoted to what occurs during prenatal and early life. And I'll go into this area more in depth because this is, and these are of this area from conception to birth. I'll go into more in depth and the first year of life. But these are all different aspects of brain development, including synaptogenesis, synaptic pruning, myelination, these type processes. And white matter development, and this is from a landmark study from 1967 from Yakovlev and Lecours. And these are all the, if you can see here, this is fetal life. This is months of the first year. So here's 2 years of age, 3, 4, and these are all different white matter tracts in the brain and when they develop. And I want you to notice here that the white matter tracts in the brain also develop mostly very early in life. Now there's studies that show that they continue to develop into adolescence and even into adulthood. And then finally, so again, another slide in which most of the events take place, major events take place in early life. And then when you look at, this is, it's going to be too small for you to read, but these are measures of gyrification. So I'm going to go into this in a little bit more detail so you know some of the measures that we're measuring with MRI. But again, you have the key thing here is here's weeks in utero and here's years. So most slides showing brain development kind of give an overview of brain development, really have much of that occurs in the brain during early life. So if we get more specifically, let's look between, so here's conception here, and here's 2 weeks of gestational age up to 6 weeks of gestational age. This is a period where these cells, there's stem cells really, neuronal precursors, they line the ventricular zone and they replicate. And between 2 and 6 weeks, they're undergoing what's called symmetric replication. So every cell becomes an identical cell. So when I was a child, my father said, for my allowance, he said, I can either give you 1 penny and double it every day for a month, or I'll give you your allowance of $2, whatever it was then. And so at a certain time in my cognitive development, I realized that if I would have said, yes, give me a penny and then double it every day, that at the end of the month, I would have been a millionaire. He was doing it kiddingly, but cells that divide exponentially have, grow tremendously, and it has to stop at some point. So this has to be under very tight genetic control. Otherwise, our brains would be extremely huge, right? So it changes at about 6 weeks. And in fact, if you irradiate the brain, so if you stop cell division during this period, the volume of the brain will change, but the cortical thickness of the brain won't change. So what happens at 6 weeks? That's when there's a change, and that's when these cells along the ventricular zone start to migrate outward to form the cortex of the brain. And it's a 6-layered cortex of the brain, and that occurs between 6 weeks and 24 weeks. So one important point to notice of this, to think about during this period of life, is that all the neurons that you're going to get for the rest of your life in the cortex are developed during this period of time. So this is, so the symmetric subdivision along the ventricular zone here turns into asymmetric cell division. It migrates out and it forms the cortical thickness. Cortical thickness is one of the measures that we measure with brain imaging. Then finally, at between 24 weeks and birth, the brain changes from a smooth brain to one that has all these fissures and folding, like we're very familiar with the adult brain. In fact, I have a slide here of how the fetus changes during fetal life, and you see at 9 months here, the brain looks very much like an adult brain, but at 6 months here, it's smooth, and there are neurological conditions, for example, lissencephaly, in which children are born with a smooth brain. There's also something called polymicrogyria, in which there's abnormalities in the gyri of the brain, likely related to abnormalities in neuronal migration. We can measure, and I thought I'd give an example of how we measure gyrification in the brain, because you can see this is an MRI of the brain. This is the cortical thickness of the gray matter on the cortex. That's something that we can measure. The white matter and the connections between different white matters, we have different ways of measuring that as well. One is using some process called diffusion tensor imaging. And we can also segment the brain into cerebrospinal fluid, white matter, and gray matter, which we do to get specific measurements of the brain. But we can measure how this curvature is. And if we take, this is a coronal section of the brain. And these are coronal sections of the brain. And Carl Zillis, who's in Jülich in Germany, came up with what's called the gyrification index. That is, if you take the outer layer, this red line is the outer layer of the brain without going inside here, and that goes in your denominator here, whatever this length is. And then in the numerator here, the top area, is the length going into all the sulci. And when you do that, you get a gyrification index. And for humans, it's around 3. This is a rodent, and you can see very little gyrification in rodents. And actually, you can look at this across phylogeny. So you can look at developments across different species, compare it to old world monkeys and apes here, catarrhines, and you can compare it to prosimians. So you can see, and this is, here is the front of the brain, and you're doing coronal slices from the front of the brain to the very back of the brain here. And what you see is that there, especially between the prosimians, considerable differences in gyrification of the brain. And for humans, it's primarily in this frontal region, the prefrontal cortex, a very important area that we hear a lot and is associated with psychopathology. So the gyrification index that I've shown was 2-dimensional, but we've become more sophisticated, and we can create triangular isosurfaces from the brain and now measure gyrification using 3D. So this is the background that I'm giving on some of the aspects of neuroimaging of things that we're measuring. And now I'm going to go to talk about, first of all, where are we studying? What is the population? But tomorrow I also give a talk where I talk more specifically about the Generation R cohort. So I'm just only going to show you 2 slides of that cohort to just give you a background. So the Generation R study is an epidemiological study of child development in Rotterdam in the Netherlands. Nearly 10,000 mothers and their children were recruited between 2002 and 2006. The inclusion criteria into this study was mothers had to be pregnant or expecting mothers, and they had to live within this area, and that's it. That was the only. About 60% of the mothers participated in the study, which is actually quite good and is reflective of the population of Rotterdam, which is actually quite ethnically diverse. You can see 58% are Dutch and other European. About 1 in 10 is in the Netherlands is Turkish. And I can see that when the Turkish football team plays a game. I see more Turkish flags flying in the city of Rotterdam than Dutch flags. So it's very ethnically diverse, and this is one of the goals that I'll talk about tomorrow of a population-based study, is to get a population, a study sample that is reflective of the population where you live. Within my group, I have 5 major research lines, and I'm going to be talking today about the first one. Tomorrow I'm going to go over some of the other ones. That's prenatal exposure and downstream effects in offspring. The one thing that I very much like about this one research line is it has, and as you'll see, a direct translation. So imaging has a direct translation into clinical, but I'll call it public health. This has a direct application into the world of public health, which you'll see. And I'll start with smoking during pregnancy. So we all know that smoking is bad, and there's been many studies showing that there's lower birth weight and other potential physical problems. But what about issues with the brain? So as part of the Generation R study, we did, so what is the effect if your child is also smoking while you are? So we start with prenatal ultrasound measures. So this is the first brain measure that we have within the Generation R. We did standardized assessments during the first, second, and third trimester, where we had several different measures that we collected. I'm going to be focusing on head circumference today, because that is a proxy for brain growth during prenatal life. Biparietal diameter is very reflective of head circumference, and then we also have measured atrial width of the lateral ventricle, and then you can get a measure of cerebellum. It's not really a diameter, it's more of a length than a diameter. So the first study was done by Sabina Rosa in the Generation R study. And what we found, so this is smoking and fetal head circumference. Here is gestational duration in weeks. So as you go from conception to 40 weeks here. And here's the message here. Non-smoking is the reference, so that's 0 here. Those who continued smoking had decreased head growth during gestational age, a linear decrease. However, those who quit smoking when they found that they were pregnant, there was no difference. It was just no difference between the non-smoking reference group. So there are effects above and beyond the effects that you have on growth of fetal birth weight related to brain development. So the message is, if you're smoking and you're pregnant, it can have effects on the brain, so you should stop. So let me, there was another question would be, so our first neuroimaging study started at age 6. So here's the question here. We know that there's decreased growth during prenatal life. What happens 6 to 9 years later? Do those differences decrease because there's brain plasticity and everything that occurs with life? Or is it still there? Are there still effects 6 to 9 years later? And we found, and work from Henan El-Maroun, that after 6 to 9 years, there remained differences related to cortical thickness in specific areas of the brain. And I would call this essentially a rather global finding, even though it doesn't cover the entire brain. It covers multiple areas that makes me think that this effect has a global effect, which makes sense. My general hypothesis is that if things occur so early during development, they're going to more likely have a global effect on brain development. So smoking is one. We also looked at other aspects of exposures during prenatal life, such as mothers with prenatal depression. And here we didn't find a global effect. We found more a local effect in prefrontal regions of thinning cortical thickness, in the lateral orbital frontal cortex of the brain, a little bit posterior as well in the medial aspect, or dorsal. So I want to talk now about something where that made me really think and really wonder about this notion of brain plasticity. I was kind of raised that the brain is incredibly plastic, and that's certainly true. It has the ability to adapt to many different events or stressors and other things that take place. But after the study with folate that I'm going to talk about, I started wondering about limits. Are there limits in the brain plasticity? And I want you to take this journey with me as I walk you through how I thought about it. So first of all, can good nutrition during pregnancy promote optimal brain development? Probably that's an easy one. But what about prevent psychiatric disorders? That's prevention of psychiatric disorders. That would be wonderful, right? Well, there is evidence for it. There's evidence for it from the Dutch hunger winter of 1944 and 1945. So the place where I live in Rotterdam, during the Second World War, the Allies came over through Normandy, and they came up through France and through Belgium, heading towards the river, towards Nijmegen and Arnhem. So this whole area above where the Rhine cuts through and goes was cut off from food sources. It was under German occupation and had very little food, and there was high rates of starvation in that area, very high rates. Mortality twice the rate of normal, fertility dropping. This plane here, this is a bomber, B-17. It's not dropping bombs. It's actually dropping food because they knew that there was a dire need for food in the area, and Rotterdam is one of the places where this occurred. There was, during this period of time, actually this is the study, the Dutch hunger winter study was the one that made the connection between folate, low folate, and neural tube defects. But there's also been studies, a twofold increase in the cumulative risk of schizophrenia. Now, when I say this, I want to add a caveat, or malnutrition is just one aspect. You also have tremendous amounts of stress and other things. So I'm going to be talking about folate, but understand that malnutrition can have many other consequences that could affect, increase the risk of schizophrenia. This is one example, but there's another one that occurred in China, the Chinese famine between 59 and 61, with over 30 to 40 million deaths. And with this, there was also a decrease in fertility, an increase in neural tube defects, and a twofold increase in the cumulative risk of schizophrenia. So there is potentially a link between prenatal nutrition and neurodevelopmental or psychiatric disorders. So why folate? Well, we cannot synthesize folate, so it has to come through the diet. We know that it's associated with neural tube defects, and we know that folate demand is increased during pregnancy. So, and when you look at the different processes that folate is involved in, repair of the genome, gene expression, amino acid, myelin formation, and neurotransmitter synthesis, we can't get it from the diet, so we have to make it, and so if we don't have enough, this could have consequences. Further, you would think that folate is abundant, but if you look at the Netherlands, and here's the, oops, let me go back. Here's the average daily intake of folate in the Netherlands, and it's lower than many different countries. And one of the reasons was before the EU started, the European Union laws, it was illegal to add any substances to food products. The EU laws changed that, but during the time of the Generation R study, folate intake was inadequate in about half the Dutch population. And approximately half the Dutch women didn't take folic acid supplements during pregnancy. I hope that that has changed. So, and in preparing for this talk, I was wondering, well, how about Turkey? I'm sure most people, when I give a talk, they say, yeah. They look for where they are on the list. You know, oh, United Kingdom, Germany, we're sitting good. So I found this study, neural tube defects in Eastern Turkey, and they looked at the rate of vitamin B12 and folate, and they found that also, about low folate was also different between the groups, and they also found in this Eastern Turkey 42.3% in the in the low income compared to 15.2. So there also are, it seems like from this study, what I'm going to present data here is also relevant for Turkey. And what we wanted to do is ask the question, even in the absence of neural tube deficits, so even when there's no major neurological problems, are there subtle differences in the brain development related to low folate? Because, folate is required in so many different processes. So our first study was looking at ultrasound measures, and this was obtained also early, middle, and late in gestational age. And we had 822 with low folate levels. I'll give the number 8 nanomoles per liter from our lab, but I have a slide that shows you what happens when we change this, and 5,123 with normal folate levels. And what we found was there was 0.1 millimeter less growth in the head circumference per week compared to controls. So I did a little math. I said, okay, if the mean third trimester circumference is 283.5 millimeters, I have that from our data. And if you look at the cortex, there's about 28,000 to 49,000 neurons per millimeter cubed, and about 1,000 to 10,000 synapses per neuron. And if you consider the cortex is approximately 2 to 5 millimeters thick, and you know that the gyrification index is around 3 of the brain, you can calculate that that is about 1.9 million neurons and 1.9 billion synapses less per week. Now, if you're pregnant, I'm sure you would want your growing baby to have as many neurons as possible, I think, but not abnormally too many, but because, you want enough to overcome adversities that go down, that could potentially occur. So now let's look 6 to 8 years later. And in this case, we restricted it to Dutch because diet and use of supplements differed between different ethnic groups or national origin groups in the Rotterdam study. So we wanted to ask, so we looked at, okay, just the Dutch mothers, but we did a subsequent study looking at, in our second neuroimaging wave, where we had many more MRI scans, we looked at the entire population, and the findings were similar. But we looked at high-resolution T1-weighted MRI processed using FreeSurfer, and we found global decrease. So here's normal folate, here's low folate, here's cubic centimeters. Those who had low folate had a global global brain volume, globally low brain volume. So this didn't quite make sense to me because we know from brain plasticity, it's also possible that, well, we know from brain plasticity that the brain should recover unless there's persistent low folate after birth. Then it could be potential effect of continued low folate during early life. So we found here is prenatal. So this is looking at 3 different periods in a longitudinal study. So this is the normal folate levels. Here is 6 and here's 10 years, so we did a subsequent study, and essentially we found that it remained, that the children had low brain volume consistently over time. Well, what about the cutoff? You know, I told you that we used an 8 nanomolar cutoff. What if we change that? And I want you to look at, so here's cerebral white matter volume, here's total brain volume. It really doesn't matter. What you find is here's 7, 8, 9, 10. So as you go towards a higher level cutoff of the serum level, it does increase. This is what you would expect. So it's a non-linear effect of low folate. So why don't the changes, why doesn't the brain recover from this? Why doesn't the brain catch up from this deficit that occurred early in life? So I mentioned that all the neurons that we have are developed during prenatal life. So what the plasticity takes place in the brain, this is my hypothesis at least, the plasticity that takes place in the brain occurs with the brain tissue that has been formed during prenatal life. And there's evidence from that from another study, a very interesting study, which is the Bucharest High-Risk Project, in which children in institutionalized care, in orphanages, in Romania, were randomized to either staying in the orphanage or going into foster care. This is a tremendous study because this study shut down all the orphanages in Romania. So it had a huge impact on the lives of children. But when they studied the total gray matter here, so this is care as usual. So these are the children, this is the total cortical gray matter for the children who remained in the orphanage. And this is the gray matter from the children who were randomized to foster care. And even though the children who were randomized to foster care had, especially before 2 years of age, had incredible improvement in cognitive performance, social performance. So they did extremely well. Their gray matter didn't change volume. And here is the reference group here. This is a study that shows actually very similar to findings that I just presented, but from a international adoption study. So it seems that there's evidence that having an optimal prenatal period of life is really important for downstream brain development. Oops, I'm going the wrong way. So, as I mentioned, we can't create more neurons in the cerebral cortex once we exit infancy. So these are healthy eating, prenatal vitamins, not smoking, decreasing stress. These are typical things that we can do during a pregnancy that could potentially help optimize the best brain. Now, there's not enough genetic information to code for the locations and connections of all brain cells. We know that. I mean, there's trillions of different brain cells and the connections in the brain, and we don't have the genetic information. What is there, 20,000 genes that code for different locations of the brain? So the brain actually works by overproducing all these neurons and then cutting the ones that are less important, called Hebbian processes. So that, in essence, has a stochastic nature to it, a chance nature. But fortunately, with the redundancy of the billions of neurons and trillions of synapses, it nearly always works out that important connections are made in the brain. But having fewer neurons or synapses could increase the risk for downstream neurodevelopmental problems. And perhaps that's the reason why we see increased risks of schizophrenia, for the Chinese famine and the Dutch hunger winter. So I did write this in a paper with a fun title. You can't lose it if you've never had it, meaning a neuron, but it's better to have it and lose it, Hebbian processes, than never to have had it at all. So I wrote that in the JCAP in 2019. So now I'm going to switch a little bit for the last few minutes to go on to autism spectrum disorder and kind of some more traditional epidemiological studies, and then an epidemiological study with a twist. So Generation R study has had a number of early life or prenatal measures that have been associated with the development of autism spectrum disorder. We found, even though there's a number of studies showing a link with folate, we did not find that folate intake was associated with less autistic traits. However, I will add, we didn't have measures of folate intake before pregnancy, and there's some studies that say actually it's very early in development, very early in gestation where the folate is crucial, and we didn't have those measures. There's been about 6 studies in the US showing a link between air pollution and autism. We had 4 studies, 4 populations combined and didn't find a difference between nitrogen oxides and particulate matter with autism. We did find a relationship with C-reactive protein, so potential for maternal immune activation during pregnancy. We now have a whole grant focusing on that, in which we're going to look at cytokines. But initially, we found a strong effect that was attenuated when we controlled for demographic variables. We have found increased rates, and we're one of the studies that showed a relationship between selective serotonin reuptake inhibitors and autistic symptoms, and also vitamin D relationship between autism. Of course, here in Turkey, with all the sun that you have, vitamin D I don't think would be as much of a problem as in the Netherlands, where during the winter we have so much rain. So one of the things that I was curious about, given all these different findings, is what if autism is a result of multiple environmental factors? So similar to how we think about genetics. So for some genetic conditions, there's called polygenic risk scores, which are psychiatric disorders are a combination of multiple genes that all have a minor effect. But what if we think about the same thing for environment? What if you have multiple environmental factors, each contribute a small effect to autism spectrum disorder? that would be an environmental-wide association study of autism spectrum disorder. And since we have multiple measures in an epidemiological study, multiple environmental measures, we can look at how all these different measures are related to autism. It's a data-driven approach that we might yield new environmental factors. So we're going to throw everything in the pool. We're going to evaluate clusters of environmental risk factors. We can generate a poly-environmental risk score to see if multiple environmental factors are involved. And then also by testing multiple variables, we can show both the positive and the negative results, which could help reduce publication bias. So we took about 1,000 different variables from different domains: parental health, maternal mental health, demographics. Some of it questionnaire data, some of it biomarkers, and we put it all into, initially we did a univariate analysis. So this is like your typical Manhattan plot, but this isn't, this is not genetics. These are environmental variables from different domains here. So each of the dot represents a domain. One of the problems that you have different than genetics is genetics variables tend to be independent. Environmental variables tend to be very dependent. So this is, the purple line here is Bonferroni correction, which is very strict correction. But we have multiple variables that are far above that level. But that's not because these are truly significant. It's because they're actually confounding, and you can see that here. So we're going to, if we took from 83 articles, we took the covariates, key covariates, and looked at their relationship with the social responsiveness scale, which is a measure of autistic symptoms. We find huge p-values. Here's gender of the child. Actually, that should be sex of the child. Oops. And it's highly correlated. So every variable that we have that correlates also with child sex is also going to be hugely different. So this shows that we had level of confounding. So what happens when we put these variables as covariates into the model? This also shows the confounding here. So these are our key variables. You can see, and these measures show how they relate to other variables here. And you can see that, they're not independent in any case. There's strong connections between many of the different variables. So when we use correct for that, correct for those adjusted for these major variables, this is still the Bonferroni. We still have a number of variables above the Bonferroni correction. Most of them were actually questionnaire data, things like obsessive compulsive symptoms. I want to add something very important. These measures were collected prospectively during prenatal life. So before we even knew that a child was autistic, these were collected, and around the time of birth, so perinatal complications. But all of these questions that were highly significant were questions, came from the questionnaire. There is also the possibility of bias related to shared bias variance. So if you have the mother reporting multiple things, they report things alike, that could play a role. So this means we could perhaps develop a mechanism to predict which children are going to have greater autistic symptoms. We did have several biomarkers, which are maternal fatty acids that were above, and a placental growth factor. The maternal fatty acids has also been reported in earlier finding, and the placental growth factor is something that we can follow up on. Now, in the last slide, I'm going to show you a hypothesis. What if we have on one axis a poly-environmental risk score? Multiple environmental factors, each contributing a small amount. On the y-axis, we put our polygenic risk score for autism spectrum disorder. Then what you would expect is, as both the polygenic risk and the poly-environmental risk go higher, you'd see more children with autism. So that's the hypothesis. So how does our data look in this? So the red dots here are the children with a diagnosis of autism spectrum disorder, and the blue are the controls. You can see that it's kind of hard to separate the red from the blue, but if you have a good imagination, you could draw a line and say, look, if you're either really low on the polygenic risk score or really low on the poly-environmental risk score, then the chances are really low that you have autism disorder. If you're above a certain area on both, then the risk is increased. So to summarize, I sit in this area of the merging fields of epidemiology and pediatric neuroimaging, and they can directly translate into the field of public health. So early exposures can influence long-term brain development, and it has an important public health message. And that important public health message is prenatal life is critical period for brain development, because you develop all the neurons that you have for the rest of your life. So let's give babies the best chance to be successful. I do not do this work alone. I do it in a team with faculty, of which I've mentioned the work of Hannah Nell Maroon, who was my postdoc, a large group of Team for Generation R collaborators, and then PhD students who are quite active. And I'll have my team wave to you. Yeah. So, thank you very much. Is there time for questions? Or? Cutting into your coffee time. I see it.
Thank you very much for your interesting and wonderful presentation. Buyurun. Burak Hoca.
Thank you. Oh, sorry. Thank you for your very generous presentation. Could prenatal environmental exposures has a selective effect on gender, particularly on fetus or embryo, male or female gender affected by these environmental exposures selectively?
Yes. So there are sex-related differences in the effects, and so some of the questions. So one is, why is autism spectrum disorder occur 4-to-1 in males compared to females, whereas when you look at later things such as mood disorders, that occurs more predominantly in females. But we are looking at specific effects now for maternal immune activation during pregnancy. And there are different factors related to, for example, placental function that could potentially play a role in some of these sex differences that we're looking at. So sex differences occurs very early in differentiation. We didn't find any sex differences related to, for example, folate. And probably what the thought is with that is that these events that occur very early in life that have global effects on brain growth, the genes involved in brain growth are similar for males and females. And but there's still a lot of questions in that area. Did I answer the question? So the other thing I just wanted to say quickly, I'm here most of the time, so if you see me, feel free to come up and ask me questions when I'm wandering around. I'd love to meet some of the Turkish, especially Turkish child and adolescent psychiatrists.
Thank you.
Yeah.
I'm wondering the effect of hormonal environment in utero, especially for estrogen or testosterone. Does this hormonal environment has selective effect on neurogenesis or neuronal surviving? Would you say something about, or is there any evidence about this hormonal environment?
Yeah.
Thank you.
So interesting. So there's one gene, the SRY gene, that when it's present on the Y chromosome, you get a male, and when it's absent or if it's not working, you get the Eve process takes place, and you have a female. That cascade sets a number of processes in action. So it's just one gene, but then there's autosomes, and then there's a hormonal difference that actually is what drives the sexual differentiation, including sexual differentiation of the brain. So much of the brain is the same, but there's certain areas that are different. And that occurs approximately 3 months, and that does have an influence on overall growth and differences in the brain, occurring early, and then, puberty. That's another area of life where there's many studies looking at that. A lot of mixed findings during puberty, but during prenatal life, there are global differences in terms of brain growth and the masculinization and feminization of the brain.
Any other questions, or I will ask that?
Thank you for your presentation. I want to ask, in your slides we see the maternal age as a risk factor, but also paternal age we know is a similar risk factor. What you think about this? It's similar risk factors with maternal or paternal, or they are dependent other factors like childhood of mother, childhood of paternal, and also education status. This is a independent risk factor, age or not? I want to ask. Thank you.
Thanks for the question. It's actually all these questions are very interesting. So we actually have just looked at parental age with respect to autism spectrum disorder. So we were looking at, to give you one example of how we're thinking about this question about paternal age. So if there's a higher risk, it's been shown that parents who are older have an increased risk of children with autism. But that could also be because there are genetic factors that if you have autistic symptoms, you just wait until you're older before you have children, because it's harder to, perhaps more challenging to negotiate all the social aspects of finding a partner, so to speak. So that's one possibility. The other possibility is that parental age, for example, there's a higher possibility for de novo mutations to occur if cell division keeps going on and going on over time, which could be associated with greater paternal age, for example. One of the challenges is if you have an older mother, you generally have an older father, not always, and vice versa. I mean, that they're correlated with each other. And men who marry much younger women also, that adds an additional bias in terms of in the equation. So what we found in terms of autism spectrum disorder was that that there is it was both. So there's a risk of, so we did find a relationship between older age of mother and father to increase risks of autism spectrum disorder. But it was also related to, so we did a theory of mind test as well, and it was also related to the theory of mind test. What was interesting also was we also found a relationship with very young mothers having higher risk. So it was like teenage mothers also had a higher risk. So we found this inverted U shape related to, with autistic symptoms. So there is a relationship, and it's probably not as simple as what, it's a combination of factors involved in finding a mate, and that just because of certain characteristic traits, but also there seems to be effect of older age on certain things. But we see that with Down syndrome and other disorders as well.
So any other questions? Then may I ask one? I'm here.
Oh,
yeah. This is about the research directions maybe. We are talking about the vulnerability for genetic and environmental factors. So I wonder if it's about a specific disorder, or is it about a cloud? Like, should we think about a tendency for neurodevelopmental growth, or specifically for autism, ADHD, or something? I mean, if it's a vulnerability or it's an exposure in utero, then is it the same thing happening for ADHD and ASD? So for our research, should we think together or separated?
Well, that's a challenging question because I go back and forth on that question. I could answer it, one day I could answer it differently than another day, depending on things. So let me tell you what I'll say today, and then you can ask me again tomorrow. So genetic factors, there is an overlap in different genetic factors of susceptibility, but there are also genetic factors that do not overlap. So there's studies, for example, looking at overall genetic susceptibility to psychopathology in general. So here's how I think about it. You have a genetic susceptibility, then you have other genetic factors that push you one way or the other. So, but I think you're gonna say, but could it be pushed in a different way if there's other environmental factors? I don't know, except that based on our studies of environmental factors, the influence of environmental factors are rather low. They're rather small compared to the genetic effects. So the odds ratios from many of these studies are 1.2, 1.5, maybe up to 2. Whereas genetics, if you have an identical twin, it's 50. So huge differences in these odds ratios. And so twin studies, for example, in autism, but also ADHD, twin studies show that genetic plays a huge role. Then environment can be broken down in the twin studies into shared and non-shared environment. And it's actually this non-shared environment that contributes most rather than the shared environment. What we don't talk about in this non-shared environment is the role of, and I alluded to it and I talked about it in the paper that the one with the fun title that you can't lose it if you've never had it, is that there are chance events that occur during development. There are, that the brain is diverse. It was meant to be diverse. If it weren't diverse, evolution would never take place. So you have this, and this is how I think about it, you have this diversity of brain development that usually goes good, but sometimes it goes awry, sometimes it goes bad, but sometimes it goes really good. And then you have a genetic advantage of some kind, and then things go on maybe in that direction. So coming back to your question, there's genetics, I think, that drive both of these things. I think environment plays a smaller factor, actually rather tiny factor, and I think that just pushes someone over the edge maybe. They're genetic susceptible, gene by environment interactions. And then you have chance factors that can also collaborate. So, yeah, I think you can ask me tomorrow. So,
any other questions or? So it was lovely to hear from you all this informative presentation. So thank you very much, and we hope to see you again in the Congress.
Thank you.
Thank you very much.
It's my first time in Turkey, so
Yeah.
thank you.